Okay, let's start off with content from last time. Questions? Any questions from the homework questions, or you're going through and you were like, I don't know what this means, I'm not sure. Fairly straightforward, people are cool with that. Okay, so for those of you that came in a few minutes later, or people at home, I don't think you guys were up here. Oh, sorry, Mom. I'll grab you right there. For those of you that weren't here, okay, for the assessment for the end of this unit, we're gonna do on Wednesday, and we'll do this during Learning Block 3, a quiz. So everyone will be at home for the quiz, including the people that are present here right now. Like, you're gonna be at home. It will be during Learning Block 3. And I guess it's technically open book. I have no way to regulate it for the people that are at home. So use whatever resources you'd like. I am gonna make it a little bit harder because it's open book, but I'll leave it open, okay? You can look at whatever resources you need to, but it's gonna be a 30-mark multiple choice quiz, okay? That covers everything from the unit, from Unit 1. I'm gonna evaluate your vocabulary lists, so those are gonna be due on Wednesday as well. You guys forget about that? Don't forget about the vocabulary list. And then what was the last component? Oh yeah, and we're gonna do a project that is a summative project that includes a bunch of stuff from the unit as well. So we'll talk about that project tomorrow, and I'll give you a little bit of time in class to work on it tomorrow. And then we'll figure out what timeline for that, but yeah, the turnaround will be fairly quick. I think we're either gonna do sort of like a short presentation, or you guys are gonna prepare a virtual poster about on your topic. But it's gonna be about a species. So you're gonna choose a species from any kingdom. I usually make people choose something that's not an animal, because you're the most familiar with animals. So I want you to go a little bit outside your comfort zone and pick something that's not an animal. And then you're gonna do a little bit of stuff about that topic. You're gonna figure out what kingdom it's in, which phylum, which class, tell me why it fits into those. You're gonna create a dichotomous key to identify from different members of that kingdom. Stuff like that. Okay, so it's fairly straightforward, and it'll make a lot of sense once we go through it. And then that's going to be the way we evaluate this unit. I think in the future, some stuff that I'd like to start implementing is like little quizzes that we do at the end of the day, but formative. Okay, so you do like a little quiz at the end of the day that helps you sort of like wrap up all the stuff that we've covered and like see if you like picked up on all the important points from the day. And not for marks, but just something so that I think I'm going to try and work those in after today. Just to make it a little bit easier for you to keep track of like what you're supposed to be getting out of every day. And then that way also the unit quiz won't just like hit you over the head and be like, what? Like, we were not used to this level of question all of a sudden. So, and maybe I'll pull some of the questions from those quizzes and put them in the summative quiz. So anyway, that's my general strategy. That means that we're gonna do sort of like 5 major projects, 1 per unit. I'm still not 100% sure on the exam. Did I talk about this already? I can't remember. So right now we're supposed to have an exam, question mark. We haven't really discussed this as a department. Obviously this course usually has an exam and there's an exam period, but we still don't have any information about how exams are going to run and how they're being scheduled, or whether we are even supposed to have exams. So like some of the resources I've gotten from the board have suggested to like move away from exams, but they haven't confirmed any of that or really given us any follow-up information. It's been very vague. So I don't have an answer for you really about how the exam is going to be. Maybe we will have one and maybe we won't. I would guess that we're moving away from exams, but I don't want to like put a guarantee on that. I'm not 100% certain. And certainly if the department and the school decide that they're going to run them for these courses, then I kind of have no choice. Then an exam it will be. But it'll definitely be a different exam than we typically write. The timeline here is still extremely rapid. It's a lot of information with big gaps. So it's like you learn a ton of stuff and then I don't see you for 2 weeks. You completely forget about this course. And then we come back and then it's really hard to have a really intensive exam without lots of review time and like time to process all the information from the course. So it will be a different type of exam. I'm not just gonna like nail you with the regular 3U exam, which is quite a intense exam. So we'll figure something out. Okay, I just want to let you know like this is all this stuff is being considered. Okay, so far in the course, we have been talking about, in general, diversity, how to assess diversity in nature, how to figure out what it is you're looking at. We talked about dichotomous keys, our general systems of classification, which is called taxonomy, and then using genetic information to enhance that system of classification, which is called phylogeny. And then last time, we went over just the major groupings of taxonomy, which are the kingdoms: bacteria, archaea, protista, fungi, plants, and animals. And you guys did a little summary here. Over the next 2 days, we're going to go into these in a little bit more detail. So today, we're going to cover 3, hopefully. Bacteria, archaea, and protists. And we're going to talk about one that doesn't appear here because we don't have a way to classify it. And we'll talk about why, but we're going to talk a little bit about viruses today too. So viruses don't fit into this system because they don't have any of the characteristics of living things. So they're not really technically alive. So they don't fit into the kingdoms of living things because they're not living technically. So anyway, we'll discuss that a little bit and about how classifying viruses. It's a different system that we use. We have talked a lot about relatedness so far in this course, and this idea of phylogeny is like how things are genetically related and like when things branched off from an evolutionary perspective historically. With viruses, we can't do that because viruses don't replicate the way other organisms replicate. So we don't know when they first appeared. There's no fossil evidence for viruses. We don't know really anything about the history of viruses, except for what we've observed and have recorded history for, and that's it. So in terms of like figuring out how things are related to other things, it's very difficult to do. We can still look at genetic differences. So we can look at, for example, somebody has, I saw a really cool YouTube video about this. Somebody got a viral infection in France, and what was it? I have to go back and look this up again. And they figured out that somebody in Great Britain went to the Caribbean, got this viral infection, went back to Great Britain. They had a flood. So the sewage from this person in Great Britain didn't get processed properly and got swept out to sea. It was eaten by mussels. And then a number of people in France ate those mussels. They were exported to France. They ate those mussels. It was hepatitis A. That's what it was. So then a number of people got hepatitis A in France, and they genetically connected it to this one couple in Great Britain who had hepatitis A in Great Britain because they genotyped the virus for both of them, which is really cool. So they figured out that, it's also super gross. So they figured out that like the fecal matter from this one couple got swept out to sea in this flood, and that all these people in France got hepatitis A from them, like directly from them. It was their hepatitis A virus because there was very few genetic mutations between their virus and the one that the people got in France, which is cool. It's cool that we can figure that out now. It's super gross, but that is cool though. So we can do some stuff with genetics, but anyway, okay, let me keep going here. So no one has any questions from the basic stuff from the 6 kingdoms? We're okay with this? No one at home? Okay. I'll try and keep an eye on the chat here, okay? I realize you guys are on just a touch of a delay. I think it's like 5 seconds, so it makes it a little bit harder, but all right, let's start with the prokaryotes. Can you guys remember what makes something a prokaryote? What is a prokaryote again? We're gonna talk about those two to start. Do you remember?
Doesn't have a cell membrane.
Oh, it does have a membrane.
It does.
That's the one thing it does have on the outside of the cell. It's missing another type of membrane.
Whoops, that's wrong.
You're like almost on the right track here.
It doesn't have membrane-bound organelles.
Right. It doesn't have the membranes on its organelles. So it's got the outer membrane to hold everything in, but it doesn't have any of the inner membranes. So all the inner membranes are gone. Just put that little reminder here. So this is no membrane-bound organelles. So originally, we thought that this was one grouping, one kingdom, and we called it Monera for a really long time, until we saw some genetic evidence and some biochemical evidence that actually showed that there are two very different groups that don't have membrane-bound organelles. And so these are the two that we're going to talk about first here, which are Eubacteria and Archaea. Eubacteria is a really, in my opinion, confusing term, because Eubacteria is not a eukaryote. So I don't know why it's called that. Eubacteria is a prokaryote. And that seems to always throw people for a loop for some reason. I don't know why the U is in front of there. Oftentimes, by the way, this group is just called bacteria. Okay, so you can call it bacteria as well, but you'll see in the textbook as Eubacteria. They kind of jump back and forth. Okay, but it's the same grouping. So that we got Eubacteria and Archaea. Some general things about these guys. First of all, we're going to talk about the roles. So prokaryotes do all kinds of niches in nature, all different kinds of stuff, and they're everywhere. Pretty much anywhere where there is an atmosphere, well, they don't even need an atmosphere, I guess, technically, depending on their type of metabolism. So, I mean, you can find them everywhere. Upper atmosphere, super deep in the Earth's crust, side of a volcano, boiling water. Like, it really doesn't matter where you go on Earth, you're going to find prokaryotes living pretty much everywhere. You're covered in them, your desk is covered in them. Although there's probably fewer on your desk because I do wipe them down with alcohol every day, which blasts the cell membranes apart. But there's going to be like dust and stuff settling that has prokaryotes in and stuff. So it's not going to be totally sterile. Anyway, they're everywhere and they do all kinds of jobs, which we're going to get to later. They are decomposers. They often produce nutrients in different environments. They can be pathogens, so they can cause illness. They could be the producer in a given environment doing photosynthesis. They do all kinds of things. Okay? And they are by far the oldest living things. They were around first. So the single-celled stuff are the first. Like I said, there's two main groupings. The first one is bacteria. And the main physical difference between bacteria and archaea is that bacteria have peptidoglycan. So peptidoglycan is a sugar protein molecule that is found in the cell wall. Bacteria have cell walls, similar to plants, although the cell walls are not rigid geometric shapes like they are in plants. So plants have, you guys have seen them under the microscope, right? They've got like sort of squarish cell walls going on. So bacteria don't have those. They can have all different kinds of shapes for their cell walls, and they're not quite as rigid, but they do have cell walls. And bacteria produce asexually, as do archaea, by binary fission. And then we're going to talk about these two terms in a minute, which is conjugation and transformation, which are the two ways where bacteria get new iterations of their DNA. That's how they change from generation to generation, so they're not always the same. And archaea don't have peptidoglycan. That's a major difference. And they often live in extreme environments, and we're going to talk about that in more detail too. So where do you find archaea? You're not going to find bacteria growing in boiling water, but you do find archaea growing in boiling water. So they are like functionally in different places usually. All right. Oh, for those of you guys at home, and I guess for the guys in the class too here, really quick, let me summarize what we're going to cover today. Probably should have started with this. I'm still getting my bearings with this. We're on day 3 of diversity. Note that any textbook chapters you need, if you're at home and the online textbook isn't working, I still haven't heard back from Nelson, which is annoying, but all of the textbook chapters are posted right there. Same thing for you guys if you don't have your textbook or don't want to take it out. They're all there. We're going to start by talking about prokaryotes. There's a really cool video here on prokaryotes that goes into a little bit more detail on the different types and how to categorize them. Question 4 in your notes you're going to do, and then there's some analysis questions. That's for the first thing that we're going to do together. I'll give you some time to work on those analysis questions. Then we're going to come back towards the end of this block and start viruses. We're going to go through a little note together on viruses. There are 2 really cool videos here on viruses. One on basic structural stuff about viruses, and the second one is about where new viruses come from, because obviously viruses appear. We see new viruses that we've never seen before. We're experiencing a pandemic with a brand new virus we've never had before. If you just note, this one does discuss coronavirus a lot, so if you're feeling sensitive about that, no problem, but just be aware that that video goes into that a lot. There's not too much homework questions from that. And then at the end of Learning Block 2, we're going to talk about protists. So that's where we're headed today. There's a little lesson with that. There's a little summary video up here about protists. It goes into a little bit more detail about the groupings of protists. And then we're going to do what we would normally have done as a lab. So you would have looked at 3 different protists under the microscope. I can't even get the protists right now, so I'm not sure what's going on. There's some kind of a shipping problem. They come from the United States, so I wonder if there's like some kind of a shipping delay. But I can't even get them. So even if we could do them, I can't somehow order them. Plus, I don't know how we would do it anyway. Maybe I would do it as a demo somehow, or if I can get them later, I'll get the microscope out here and show you what they look like. And then there's some analysis questions that go along with that. Okay, so that's what we're going to cover today. Let me keep going here. And then the last block, obviously, is time for you to complete any questions from the day and come and visit. For all of those of you that did come to visit, thank you. One more thing I just want to mention really briefly is that I'm not available by email after 8 p.m. Okay, I'm still getting a lot of 2 in the morning emails. Don't take this personally, guys, but I don't want to hear from you at 2 in the morning. Okay, just you can schedule an email to send at 8 a.m. the next day. Just do that. Okay, don't send me an email at 2 a.m. unless it's an emergency. Okay, I'm getting like a lot of them, and it's from my other class as well. But please don't send me emails at 2 in the morning. No, thank you. I'll pass. Okay. Single cells. Yeah, they don't have organelles. They're obviously the smallest because they're single-celled, and their cells are very small. So do you guys in grade 10 look at some things under the microscope, right? You would have looked at some kind of cells in grade 10, hopefully. Yes, something? Okay, so if you looked at some kind of cells under the microscope, bacterial cells, even on high power. So if you were to look at a bacterial cell on a high power under the microscope, it would look like that. Okay, at 400 times magnification, it looks like a dot. They are very small, much smaller than eukaryotic cells. So that's the first thing. They're extremely tiny. You can resolve them with a light microscope, but just not these ones. So you need like a nice, powerful light microscope to actually see what's going on inside. Something with at least 1,000 times magnification. Like I said, they're found everywhere. Surface, subsurface, water, soil, atmosphere. I mentioned boiling hot springs, ice, high salt environments. Those are mostly Archaea that do that. And you have about 100 trillion living on your body, doing all kinds of things. Regulating pH, eating your dead skin cells. Oh, all kinds of stuff. Some of them are probably pathogens, but you actually don't really have too many pathogenic bacteria living on your body. And most of them are being regulated by the conditions that are on the surface of your skin. So we probably have only identified about 1% of the prokaryotes that are out there. Like I said, every time we take a soil sample, I think I mentioned this on Friday, we always find like 100 new ones. It's just like, oh, what's this? What's this? What's this thing? There's just, oh, I've never seen this DNA before. So we really don't know probably the majority of prokaryotes. One thing humans are really good at is identifying the pathogens. So we're very good at figuring out what makes us sick because that's an emergency. We have to figure that stuff out. But a lot of the other prokaryotes that don't make humans ill or are not involved in human health, we don't do a great job of figuring out what those things are and cataloging them because it's just not as pressing for us to figure that stuff out. So about 1% is a good estimate of what we currently know. So what are some examples of things that prokaryotes do? Well, pathogens is the one that is probably the most concerning for us. Stuff that's caused by bacteria, bubonic plague. We don't have too much of that around anymore, although they recently had an outbreak in Madagascar of the bubonic plague, but it's fairly rare. Luckily, it can be treated fairly easily with antibiotics. So you can. Too bad the people in the Middle Ages didn't have that, but you can just pretty much just treat it with antibiotics now. Bacterial meningitis is caused by bacteria. You can get food poisoning, E. coli. I think I mispronounced this last time. I'm really terrible at this word. The E in E. coli is Escherichia? 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 I'm going to look this up again. I think I said Escherichia last time, but the word is, I'll throw it up on here because this word always messes with my head. It's Escherichia. There we go. Whoa, what a word. Escherichia. There we go. No wonder we just say E. coli. That's a real mouthful. Anyway, causes food poisoning. You've got a ton of it growing in your large intestine, but not your small intestine. If it gets back up there, you're going to get yourself into trouble. Salmonella, again, causes food poisoning. Leprosy is caused by a bacterial infection. Leprosy is kind of a cool one because it's a really, really tiny bacteria, and it can live inside of your own cells. So it operates in some ways like a virus, but the bacteria grows inside of your cells, which is kind of cool. Although it's not cool if you have it, but tuberculosis is caused by a bacterial infection. So all these things are bacterial. So there are pathogens. There aren't a lot of archaea pathogens, by the way, which is kind of interesting. So it's almost exclusively bacteria that cause illness in humans. There's some decomposers. These are really important for ecological reasons. They break down all types of nutrients, proteins, carbohydrates, fats, structural proteins, all kinds of stuff from nature, and allows them to be recycled back into the food chain again. They are often producers. That is, they form the bottom of food webs. They do photosynthesis, although like a modified version of photosynthesis. They don't have chloroplasts like plants do. So the way they do photosynthesis is a little bit different, but the end product is the same. They produce energy molecules from light, and they produce oxygen. So they remove carbon dioxide from the atmosphere. So the end product is the same. So that's kind of important. Prokaryotes do the majority of the photosynthesizing. So it's weird to think of that, but there's more prokaryotes doing photosynthesis, like quantity-wise, in terms of removing carbon dioxide from the atmosphere and producing oxygen, than there are plants. We don't really talk about the prokaryote component of that, but this is the most important component of it. More is happening in the ocean through photosynthesis through prokaryotes than is happening on land through plants. So it is kind of important. You guys talk about nitrogen fixation in grade 9, probably? Nitrogen cycle? Nitrogen comes out of the atmosphere and is turned into ammonia, and then other bacteria take the ammonia and turn it into nitrite, and then some of them turn it into nitrate, and then the plants take up the nitrate and make protein out of it. Does that sound familiar? So a lot of that is done by bacteria. Produce ammonia, nitrate, and nitrite, which is absolutely essential for plants. So if we removed the bacteria from that nitrogen cycle, there is no bioavailable nitrogen for plants to make protein. It breaks the natural cycling of nitrogen without bacteria. That's why when you're growing crops or you are managing a forest or something like that, often a big part of your job is monitoring what is growing in the soil, what prokaryotically is growing in the soil, because those soil bacteria are a huge part of the reason you have a healthy ecosystem. You're on a farm, you can't have bacteria-free soil. If you bleach your soil and lose all the bacteria from your soil, you're going to lose a big part of the bottom of the food chain here, especially the nitrogen component. We can also engineer bacteria to produce extra nitrogen for plant roots. So that's like an area of research. People work on those types of bacteria. They're called rhizobium. So a lot of people are into rhizobia. You can engineer them to produce fertilizer basically for plants. They go right on the roots of your plants. You have to use less fertilizer. It's kind of a cool system. And then lastly, you have a bunch in your digestive tract. Some of it's bacteria, E. coli, and a few other types of bacteria, but there are some archaea that live in your gut as well. They produce methane. They're methanogens. So that includes the production of vitamin K and vitamin B12. So those are actually fairly rare in your diet. There isn't a lot of those two nutrients typically in people's diets. So the bacteria in your gut make those, usually out of long chain carbohydrates that can't be broken down in the upper part of your digestive tract. It makes these nutrients out of the stuff that's at the bottom. And then you absorb it through your large intestine. So we actually require these in our gut. When you take antibiotics, for whatever reason, you usually kill all of those, which is problematic. So if you are taking antibiotics regularly for whatever reason, you have to build back up your populations of bacteria in your gut so you can continue to take these vitamins up. And there are a number of inflammatory illnesses of the bowel that are caused by taking antibiotics, which removes basically all the prokaryotic regulators that exist in your gut. Yes?
How long can you go without vitamin K and B12?
Good question. You can be deficient in those, and there are symptoms of being deficient. I don't know, though. I don't know how long. I don't know. That's a good question. I have to look into that. I don't know off the top of my head. I do know that the inflammatory illnesses that you get from having no bacteria in your gut or not enough bacteria in your gut are like quite serious. And one of the ways to treat things like inflammatory bowel illnesses is to take somebody else's gut biome and put it in someone else. It's called a fecal transplant. This is when you take a little poop from somebody and you put it in somebody else. Which sounds really weird, but it works really well. It's extremely awkward conversation for you to have with your doctor. So it's very underprescribed, even though the efficacy of it is really good. If you look at the research behind it, it's a very efficacious treatment. It works well for a lot of inflammatory bowel illnesses, but it doesn't get prescribed very often because doctors don't want to tell people that they need to get a shot of somebody else's poop in order to fix their gut problem. So it doesn't get utilized very often, but it does in fact work quite well for a lot of inflammatory bowel illnesses. So, true story. Okay. Oh, sorry. Yes, I can remove that logo. Oh, I didn't realize that was in the way. Let's just make it nice and small, put it in the corner. And lastly, we can use it to make stuff. Yogurt, for example. Cheese. You need bacterial cultures for both of those. The exact species are slipping my mind here that we use for yogurt and cheese, but anyway, both of them are bacterial cultures. And we can use it for synthetic production of all types of molecules. Bacteria we can use basically is the 3D printer of biological molecules. It's such a cool system. Because bacteria are so simple, they are essentially just like a balloon with ribosomes and some DNA inside and a couple other small things, but really that's all that they are. They're very simple inside. Because they're so simple, you can just take a piece of DNA, usually in a loop called a plasmid, that codes for insulin. Insulin is a great example of something that we do. And you can just pop that inside a bacteria, and the bacteria will read it and just make the thing that's on that string of DNA. So it'll just make insulin for you, Or if you want to produce some testosterone, you can genetically engineer some E. coli to just make some testosterone for you, or whatever synthetic molecule you want. We use it for drug production, but insulin is the big one. Does anybody know how we originally got insulin to treat diabetics? This is actually kind of sad, but no. So we used to use dogs, and we used to tie off the pancreas for dogs until it essentially like died. And then you would just extract the insulin from the pancreas that we tied off. We used to use dog insulin because it's very similar biochemically to human insulin. But that's a terrible way to make insulin, guys. That's horrible. And also you can't make huge quantities of insulin that way, not without like a bunch of unpleasantness. So a much better way to make insulin, and this is like a groundbreaking process, is we just get bacteria to make the insulin for us. We found the gene for insulin making, popped it in some E. coli, and then we just grow big vats of that E. coli. We give it some food, and it just makes insulin for us. We just extract the insulin at the end. So we can do this with all kinds of biological molecules. We can get bacteria to build them for us, which is a really cool process. So they're very useful. Okay, let's talk about what they look like. This is a little bacterium right here. Now keep in mind that they're not all going to have the exact same shape or external structures, but this is a general model of a bacterium, okay? In general, they have these things. In the middle, I'm trying to figure out where to stop here, where to start here rather. They have some DNA. Gotta have that. All living things have that in the middle, even some non-living things like viruses. You gotta have some DNA. That encodes for the building of all the different pieces of the bacterium. DNA do not have introns. So introns are sections of DNA that don't do anything. They're just like extra. Extra DNA in bacteria really doesn't exist. That's one way to be able to tell bacteria apart from archaea. Archaea, while they look exactly the same on the outside, archaea have introns. They have extra DNA that doesn't code for anything. You have extra DNA that doesn't code for anything, called introns. Okay, we used to call it junk DNA, but it's not, that's a bad name for it because it does have a function, but it just doesn't code for things. Question?
What is it called again? When they have extra DNA?
Introns.
How do you spell that?
I'm going to add it right here. Loops of DNA that regulate cell function. No introns. This is non-coding DNA. So again, that's specific to bacteria. Archaea do have introns. There's also little loops of DNA inside bacteria called plasmids. So they come in hula hoops of DNA. They're usually pretty short, and they often only code for one single thing, for one protein. And these are cool because bacteria can exchange them. So if there's like two bacteria and one of them knows how to make a certain protein, it can sidle up next to another bacteria, create a little tube between the two of them. Does this one have a tube? No, it doesn't. Do I have another picture with one with a tube? No, I don't. Oh, yeah, actually, I would think that these are the tubes. That's fine. It can build these. Here's some right here. So these are pili. They look kind of like hairs, but they can use those pili to poke into a neighboring bacteria and say, here, have some of my DNA. Can send some of those plasmids across to the neighbor. Okay? So the pili are used to exchange genes. So that's called conjugation. It's kind of annoying that there's multiple words for some of this stuff. When a species can give its genetic information to someone in the same generation, that would be like you giving someone else in the room some of your genes to use. That process is called horizontal gene transfer. It's horizontal because you're giving it to someone in the same generation.
Staff and students. You're going to have a walk-through fire drill, not a real fire drill practice, but just a walk-through today. If staff could read page 11 in the shared emergency folder, when you have a chance, I just want you to walk through a drill with your class and note that the wayfinder arrows and staircases should be ignored in a fire, in the real fire or for this drill. Make sure you take the time to go through the process with your students and practice when you have a chance to avoid meeting up with others in the halls. Take the students right outside to where they should be standing and waiting. And you could do this morning, this afternoon. Just check in the hallways to make sure that not a lot of people are out there because we want to maintain social distancing. Thank you.
Okay. Ring of fire bell. Fire safety zones. Reduce fire. Okay, it's just a normal fire drill. Yeah, this isn't different from a normal fire drill. The only difference is they're not going to ring the bell. So we just decide when we want to do it. So I guess we just do it later, I guess. I'll come back to this. We'll just pop out for a second and do the drill. Interesting. Okay, sorry. So that's called horizontal gene transfer when you give genetic information to someone in your own generation. Only prokaryotes can do that. Actually, hold on a second. Can any protists or anything do that? As far as I know, only prokaryotes can do that. I'm trying to think if that's protists can do that. I don't think so. I think this is only prokaryotes. And obviously, that's a way for them to increase their diversity. They can also do something where they can suck up DNA from their external environment. So if a bunch of bacteria die in their neighborhood and their DNA just gets kind of exploded into the area around them, the bacteria can kind of go through the waste and take DNA out of the waste and potentially gain new abilities by picking the DNA out of the waste, which is really weird. So they can also do that. So that's a process called transformation. I better add that in here too. Or should I put that maybe over here? So bacteria can pick up DNA from their neighborhood. Make this smaller. Ooh, slow. Pick up. Oops, I just spelled that wrong. Bacterial. Why is this going so slow? Transformation. So that's like, that's also really useful for us because if we want to get bacteria to start building insulin, for example, all you have to do is take the DNA from making insulin and just like sprinkle it around, around the bacteria. And the bacteria will just kind of say, oh, I could use this, I could use this. Now, oftentimes you have to give them a little bit of a push. You can heat it up, or sometimes you use a little bit of electricity to get them to open up and take in the DNA from the surrounding environment, but they will do it naturally as well. But they can just pick up DNA from their surrounding environment and incorporate it. So that's called transformation. We used to do a lab in this course, which I don't want to bum everybody out here, but saying stuff we used to do. But we used to transform some bacteria, and I actually think that we're going to move this lab to grade 12. So you guys probably will do it if you take grade 12 bio. But you're going to grow some bacteria, sprinkle some DNA, and then get them to do something new. The thing that we usually get them to do is glow in the dark, which is kind of cool. So it's a protein to make them glow in the dark. Anyway, I think we're moving the lab to grade 12. So that's called bacterial transformation. So they do have an ability to do something that is similar to sexual reproduction, in that they can exchange genes to make themselves more diverse through that horizontal gene transfer. They create a pili, send some DNA to their neighbor. But they don't have traditional sexual reproduction, as we're going to discuss later in the course. Okay, they do often have flagellum or something similar to propel the bacteria around. Not all bacteria have that. So some have flagella and some are just kind of floating. They don't really have any propulsion mechanism. Flagella are fairly energy intensive, so it really depends on the metabolism and the environment of the bacteria. Not all are well suited to having that. And now more structurally, most bacteria have a peptidoglycan layer, which is, like I said, a large chain of molecules that gives it some strength and rigidity, just like a plant cell wall. It has a cell membrane, which does the same thing that cell membranes do always in plants, in organisms, which is allowing the control of what goes in and out. So it has transmembrane proteins that can pump stuff into the bacteria or pump stuff out. That's a cell membrane, just like any cell membrane that you'd find in an animal or plant cell. And lastly, most bacteria have a capsule around the outside. So the capsule component prevents water loss. Unlike your cells, which are always bathed in this fluid, your extracellular fluid, which is continuous with your blood, that's a nice environment for a cell to be in all the time. Oh, it's nice in there. It's like wet and nutrients are all around you and stuff. How lovely. But bacteria don't get to have that, right? Like they could just be, they're growing on your desk, for example. That could be a very dry environment. That's tough for a single-celled organism to have such a rough external environment. So they do need like a protective layer on the outside. It's almost like a seed coat for seeds. It protects them. That's the capsule. That capsule is also used to protect bacteria from viruses. So viruses attack bacteria too. A virus that attacks bacteria is called a bacteriophage. Oftentimes when you see pictures of viruses and they look like that little lunar lander, you may have seen these before, like in a textbook or something. Those are actually bacteriophages. And they can protect the bacteria from temperature change. Again, it's a single cell all by itself, or maybe in a little group. So if things get a little too warm or a little too cold, the capsule protects them. Now if it gets really warm, like above 40, 50 degrees C, maybe 60 degrees C, forget about it. Then you're starting to get rupturing of your capsule. So, which is good for us. We like to kill them, generally. Sterilize things. Extreme cold usually puts bacteria into more of a hibernation. So it doesn't really rupture the capsule. If you take something that's full of bacteria and put it in your freezer, it inhibits their growth, so it may lock their growth, but it doesn't usually kill them. So it just kind of puts them into a hibernation stage. Some of them will die. Some of them will have a rupture of their membrane from the extreme cold, from the crystallization of the water, but not all of them. Any questions about the general structure of bacteria? That's it. I mean, I guess that seems like a lot, but it's way, way less complicated than a eukaryotic cell. You guys did cell diagrams and stuff in grade 9 and 10, right? Like there's a lot more going on in there. This is it. That's the whole thing. All right, let's talk about the general shapes that you find bacteria in. Okay, so there are 3 general shapes. For the most part, there are exceptions to this, but these in general are the shapes that you always find bacteria in. And you'll hear this in the name of the bacteria. Often they're named with part of their shape in their name. So a cocci bacteria, COCCI, is just a sphere. That's the one on the far left there. Those are cocci. Streptococci, bacteria, whatever. The ones in the middle there, the ones that are like tubes, are called bacilli. It's like streptobacilli. And lastly, Spirilla. Spirilla look like little corkscrews. What an unusual shape to find a cell, a membrane in, like a corkscrew shape, but it works for them. So they can be cocci, bacilli, or Spirilla. And they're also found in different groupings. If there's only one, I didn't even put only one on here, we call those mono. One is just like one single one by itself. That's mono. A bacteria just found by itself. If there's 2, we call that a diplo. So if it's 2 cocci together and they're found in groups of 2, that's a diplococci or a diplococci. There's some variation in terms of how people pronounce these. I'll say my profs in university couldn't agree. So if you've got clumps, a clump is called a staphylo. So a staphylococci is a clump of cocci. For example, you may have heard of Staph aureus, Staphylococci aureus. Staphylococci aureus is a very common skin infection caused by Staphylococci aureus. This is like the #1 skin infection that people get at the gym. People get Staph aureus infections at the gym all the time because if you don't clean the gym equipment properly, you get a Staph aureus infection. That's why, for the most part, why those spray bottles are at the gym. It's to prevent buildup of Staph aureus, which is extremely contagious. So you can get that fairly easily at the gym. That's why you gotta clean things up. I think that's probably not people's primary concern now, but previous to this, that was the thing that you needed to watch out for at the gym. And then lastly, you got your streptos. A strepto means a strip. So they come in long chains. And obviously you can apply this to any shape, although the Spirilla are usually by themselves. So you don't see a lot of Spirilla. They do exist, but they tend to be found on their own more. Whereas the cocci and the bacilli are often found in these other groupings, streptobacilli or streptococci. So Streptococcus pharyngitis is the bacteria that you typically see causing strep throat. You guys may have heard of this before. You may have had strep throat before. Sometimes they just call it strep. Treat that with some antibiotics usually. Okay, so those are the basic shapes. Keeping in mind that naming bacteria based on their shape and grouping is not necessarily a great strategy because you can get multiple species that you have the same shape and the same groupings. So we do use that system, but again, this is like one of those weird things where we currently understand so much more about the genetics of these things and the variety, but previous to that we didn't have any of that information, so we categorized stuff principally on what we could see. And so there's an overlap here. We have a system where we often use shapes and what we see, but also a bunch of genetic evidence which kind of overrides this a little bit. And so we've had to branch out and kind of adapt what we already knew based on the genetic evidence. So there is a little bit of confusion there, just as a heads up. Okay, another way that you can distinguish between bacteria. So shapes are one way. We used to use shapes all the time. It's what you can see under the microscope. We can distinguish them based on their metabolism. So we can say that they are autotrophs. An autotroph is something that is self-feeding. You would have talked about this in grade 9, hopefully. Autotrophs are the things at the bottom of the food chain, right? They make their own food. Troph means to eat, and so autotroph just means self-feeding, or self-eating, I guess, technically, but that means the same thing. They usually do photosynthesis. Most autotrophs do photosynthesis. There are some types of bacteria that produce food, but from complex molecules that are found on the ocean floor. Those are chemoautotrophs. They can use like lead or gold compounds or like other weird stuff that's found on the ocean floor, like near thermal vents, to do their energy metabolism. Those are quite rare. Certainly at the surface they don't exist, but that does also exist though. So there are chemoautotrophs that do that. Chemoautotrophs don't use light as part of their autotrophic metabolism. And then you also have your heterotrophic bacteria. So that's bacteria that eats stuff. That makes up probably the majority of the varieties of bacteria. All of the bacteria in your body, for example, are heterotrophs. They're mostly eating the dead material from your skin that you're not using anymore. They actually help you shed the material that you're no longer using. Trying to think about that too much. Did we mention binary division already? I feel like we talked about this. Bacterial cells just split, make two. That's how they reproduce. It's kind of a weird way to poke that into the note. I'm not really sure why I put that there. That's the reproductive strategy. Bacteria gets really big. It does something that is basically mitosis, and then it turns into two. It's quite different from the mitosis that you talked about in grade 10 because there's no chromosomes. So you don't get that like line up in the center, pull to the sides. It has like a different method for doing the same thing, but I mean the end product is the same. You get two identical cells. All right, so you could distinguish between how they get their food, autotrophs or heterotrophs, or you could distinguish them based on how they are doing their metabolism. So you could say, well, this bacteria needs oxygen. Another way to say that is it's an obligate aerobe. An obligate aerobe means it needs oxygen in order to survive. Sure. Try not to make a habit of it. We're not supposed to plug them in class, but yeah, you can use that. Or you could say, yeah, it'll use oxygen to do its metabolism, but it doesn't need it. If it doesn't have oxygen, it just does something else. It prefers oxygen. That's called a facultative aerobe. Facultative means that it prefers that, but it doesn't need that. A lot of bacteria are facultative or aerobes. So you can like put them in a jar and yeah, they'll grow, but they won't grow as fast as they would have grown if they weren't in a jar and had like easy access to oxygen. Oftentimes, when they don't have access to oxygen, they'll do fermentation. So we also take advantage of stuff like that to make alcohol. Although not through bacteria, we usually do that through type of fungus. You can also do that. Or you could also say, well, it is an obligate. It does need no oxygen. So if you're an obligate anaerobe, that means you need no oxygen to live. Those bacteria, if they are exposed to oxygen, it kills them. So that exists too. It's a bacteria for all occasions. They're everywhere. An example of an obligate anaerobe would be botulism. Botulism is an obligate anaerobe. It's killed by oxygen, so it really, really loves to grow in conditions where you've removed the oxygen. Like, for example, let's say you didn't can your fruit very well at the end of the season if you're doing fruit canning or making jam or something, and you have a little bit of botulism growing in there. Oh, it loves that. It'll start growing all over inside that thing. So then when you open up the container, then you got a whole bunch of botulism in there. I might get you guys to look into botulism in more detail later. It's a pretty interesting bacterial infection. Terrifying, but it's kind of cool though. All right, this little part I'm going to get you to do on your own. We'll come back to this later, but I'm going to get you to just look at some examples of some bacterial diseases. They're on page 51 in your text. There are a number of bacterial pathogens. I try not to focus on the pathogens because the vast majority of bacterial species are not pathogenic. They don't cause illness in humans, and they're in fact super useful. So I hate the idea, this is like an idea in the public as well, that all bacteria are bad. People are always trying to sterilize everything. But bacteria have a lot of really important biological functions, even on and inside your body. So it's important not to write them off as purely pathogenic, but you got to be aware of them. So there is obviously some pathogenic bacteria, and they do cause a lot of problems. So I'm going to get you guys to come back to that. We'll do that in a minute. Okay. I'm going to break in a second here. I've been talking a lot. Let's talk a little bit about the overuse of antibiotics. A huge pet peeve of mine and doctors everywhere, although some doctors still do this. Shame on you. We have a huge tendency to overprescribe, mostly because people always ask for antibiotics, because we have like a magic pill society where people always want a pill for every illness, no matter what it is, even if it doesn't make sense. That is a real thing, unfortunately. So the way that you get antibiotic resistance, which is a huge, huge problem. A lot of illnesses that we used to be able to treat with antibiotics, the antibiotics don't work anymore. Very problematic. And it's all because of evolution. So a basic evolutionary process at work here. Imagine a grouping of bacteria. Okay, here's my bacterial grouping. And I've used my colors to denote sensitivity to erythromycin. Okay, that is how easily they're killed by erythromycin is an antibiotic. Okay, a common antibiotic. Okay, let me explain my color code here. So I'm going to say red. Red here is very susceptible. Yellow is like a little susceptible to erythromycin. And green is resistant to erythromycin. So red is killed by erythromycin. What is going on? Why is this such a delay on here? Is it MY erythromycin? Oh yeah, it is. All right, so we got three sort of basic types of bacteria in a population. There is always diversity in the population. Just like in this room, we're all organisms in here, and there's genetic diversity among us. We don't have the same genes. Same thing is true for bacteria. So in a given grouping of bacteria, a lot of them will share a lot of their bacterial DNA. They may have all come from a single bacterium and multiplied, but they're not going to be identical. Mutations happen, random mutations happen in every generation just due to random errors in replicating their DNA. So you're going to get some variety in every population. Some are going to be more resistant to the antibiotics than others. When you treat a bacterial population like this with an antibiotic, okay? So let's say that this is a bacterial infection. There's kind of two ways this can go. First of all, there's bacterial infections that don't require antibiotics. These are things that you would just resolve on your own, okay? And there are also bacterial infections that do require antibiotics, but you don't take the full course, okay? So you don't take the full week. You start feeling better and you say, I don't need to take these antibiotics anymore. And you stop taking them. Okay, so both of those situations, you get the same end product, which is you don't kill them all right? So you take a bunch of antibiotics and you don't end up killing them all. Okay, you either took them when you didn't need to or you didn't take the full course. So what ends up happening is you're going to kill all of the ones that are sensitive to erythromycin, right? Those are the red ones. Those are all going to get killed because as soon as erythromycin is in the system, it's going to cause them to rupture their cell membrane or inhibit their reproduction or whatever the antibiotic does. It does different things depending on the antibiotic. Okay, so you're not going to have any red ones in your next population. They're all dead or they didn't replicate. But you might have a couple yellow ones. Okay, you'll get a few. They're still sensitive, so you're not going to get a lot of these. A lot of them are going to be killed, but the green ones won't be killed. Okay, so you had a population that looks like the first population here, which is largely killed by erythromycin. Okay, this is a antibiotic sensitive population. We like these. If you want to get rid of a bacterial infection, you want to be able to kill it, right? You want to have the population that's mostly sensitive. When you use the antibiotic treatment, assuming you don't get rid of all of the bacteria, what you're left with is a population that is mostly not sensitive to the antibiotic. So when this population regrows, as it inevitably will, all of a sudden you're going to end up with a population that's now 50% in this case, but it's not necessarily 50, but 50 in this case. Now it's 50% resistant. Okay, 50% won't be killed. And if you treat it again and don't use a full course, you're going to reinforce this again. So you're going to end up killing mostly yellow, and then when it regrows, it will be mostly green, mostly resistant. And every time you do it, you make the population more and more and more resistant to antibiotics. The same is true, by the way, if you just put antibiotics out into the natural environment. You flush them down the toilet. You are constantly using them in livestock that are not sick. That's something that we don't do here a lot in Canada, but in the US it's a very common practice, which is just to give every animal antibiotics all the time, just so that they don't get sick on your farm. So the problem with that is not necessarily that it gets into the food, although that there may be a concern with that as well. But the other issue here is that you're creating these huge populations of bacteria that are resistant to your antibiotics. And we do this all the time. A great example, we were talking about tuberculosis. Tuberculosis is a bacterial infection. We have created some strains of tuberculosis that we cannot treat. There is no antibiotic that treats it. They are super drug-resistant strains of tuberculosis. So when you get that tuberculosis, we don't have anything for you anymore. Tuberculosis is a terrible illness that we've been able to effectively treat for 100 years, or maybe 60 years. But now there are some cases that we just can't do anything for anymore. When you get that, we can't do anything for you. Same thing is true for things like syphilis and chlamydia, common sexually transmitted illnesses. We used to always be able to treat those. We had great antibiotics, but because we misuse the antibiotics, we're starting to get into an area here where we can't always treat those infections. There are super drug-resistant chlamydias where you're kind of stuck with chlamydia. We can't treat it properly anymore. You end up with like a long-term case of chlamydia, which is terrible, okay? So these are the real issues that we're dealing with in medicine, especially over prescription. Don't ask your doctor for antibiotics if they're not necessary. They may give in. They shouldn't, but they may. So what you've just seen happening there, a population completely changing its genetics over time, is evolution. That is exactly evolution right there. And we'll talk about this more in the evolution unit, but that is the evolution of a bacteria. You put an environmental pressure on something and said, only you survive, but you don't survive. And then at the end, the genetics of the population are different. That's evolution. So this is a bacterial evolution that is caused by humans. Not necessarily a good one. You can reverse these processes, by the way, by using fewer antibiotics. There is a reason why the red ones here were more common at the beginning. And that's because usually they're just better at surviving. The ones that are less resistant less resistant to antibiotics are usually just better at multiplying. So oftentimes when you stop using antibiotics, you get this profile coming back again. You start seeing more bacteria that are susceptible to antibiotics again. But you have to do that. You have to allow them to reemerge. Okay, so that's my little spiel here on bacterial resistance to antibiotics. We're gonna maybe get into that more later in the course in the evolution unit, but last little thing I want to mention here is Archaea. Archaea are not as common, and we don't use them for as many things. In other words, we don't use them for making biological molecules, and we don't use them to make yogurt or anything like that. But they're still interesting. I don't have as much information on them, but they inhabit extreme environments. So there are some general classes of Archaea. Again, the main difference here is that they don't have peptidoglycan in their cell wall. They also have some things that we have as eukaryotes. Their DNA is wrapped around tiny little proteins called histones. Archaea have that, and bacteria don't have that. They don't have histones. So it's very likely that multi-celled organisms like us evolved from Archaea and not from bacteria. So this is a separate lineage. They have some interesting genes that we have. So some examples of the classes of Archaea: Methanogens. They live in your gut. They're making gas in there right now. These are also like common bog bacteria that are decomposers, and they produce methane in swamps and things like that. Some Archaea live in really high saline environments. That's impossible for a bacteria to do. If you have a really high saline environment, it dehydrates regular bacteria and causes them to die. So really saturated saltwater solutions, bacteria can't live in that. That's why we pickle things, by the way. It kills bacteria and prevents bacterial growth when you pickle stuff. But halophiles don't care about that. They can live in a really high salt environment, which is kind of cool. So like on the salt flats, for example, you won't see a lot of bacteria living on a salt flat, but halophiles will live there. You got your extreme thermophiles. An extreme thermophile can live in temperatures up to 114 degrees Celsius, which is above boiling. So you can't kill these by boiling. They actually prefer to live in boiling water. If the water gets too cold, they die. Anybody been to Yellowstone National Park in the United States? No. I've been to Yellowstone, eh? Yellowstone has a whole bunch of thermal vents in it. This is one of them. It's very pretty. Looks like that. So that's a thermal vent. It's a bunch of water that's being squeezed out of volcanic rock. It's very volcanically active in Yellowstone. And those colors are produced by extreme thermophiles. They're thermophiles. So the reason that there's different colors is because different species of thermophiles live at different temperatures. The ones towards the center, those are like the green and yellow ones. They like very high temperatures. And then as you go further out, like the light and dark orange, those ones like slightly lower temperatures. But the water in the center is boiling. There's boiling water coming out of this thing. And pretty much the only thing that lives in it is extreme thermophiles. They're really pretty though. If you ever go to Yellowstone, there's a whole bunch of thermal vents like that have beautiful colors. And then lastly, you got your Psychophiles. That's the coolest name, I think. And those are bacteria that can reproduce in extreme cold environments below the freezing point of water. So they have some interesting sugar structures in their cell, in their cytoplasm, that allow it to remain a liquid and continue to reproduce even in sub-zero temperatures, which is pretty cool. And those are the kinds of prokaryotes that you find in the Arctic. There's not a lot of prokaryotic life in the Arctic due to extreme cold, but you do see these growing there. So there's prokaryotes everywhere, even in Antarctica. Okay, that's enough talking from me. I'm going to give you 15 minutes, 20 minutes to work on the questions here. Does anybody have any questions about anything we've covered so far? Just talking about basic classification information regarding prokaryotes. Two major groups, two major kingdoms, bacteria, or Eubacteria and Archaea. Okay, so what you're going to do, there's a little video for you to watch. I'll jump back to that here quickly. Wish this didn't crash so much. There's a little video here to watch, which is a summary of information on prokaryotes. There's question 4 in your course notes, which asks you to take a look at some pathogens. It's this one right here. Okay, use page 51 from your textbook. And then there are 2 analysis questions from the text. And then we're going to come back and talk about viruses. It's 9:40. We have till 10:20. Let's come back at 5 after 10. Okay, that would give you 25 minutes, right? Okay, let's come back at 5 after 10. Hey guys. Sorry, I wrote 11:05 on there, but I actually meant 10:05. I got to get the schedule figured out here. Sorry if that confused people. Okay, I'm just going to get started on the next part here. We're really briefly going to talk about viruses, I think, and then we're going to head into the break. So to get us started thinking about viruses, this is obviously different than all the other stuff in all the other kingdoms, mostly because of these things. So in grade 10, maybe you would have talked about what the factors are that make something alive, like what it does. So these are they right here. And you can do this with anything we've covered so far, any of the prokaryotes or anything from any of the kingdoms. But if you go through this list for viruses, you run into some problems. So first of all, do they respond to stimuli? So that's something that in general all living things do. If you poke them, do they respond in some way? And the answer for viruses is no, they don't. We're going to talk about like what viruses are made out of, like functionally in a second, but no, they don't. So a virus particle is an inert particle. Do they eat? Do they have a metabolism? Do they take in nutrients? Nope. They don't do that. Do they adapt to their environment? So if there's a change in environmental conditions, do they modify their protein expression? Do they move? Do they do anything? Nope. Do they reproduce? Do they make copies of themselves? Nope. Not on their own, they don't. Something else does that for them, completely independent of them. Do they produce waste products? Nope. Do they grow over their life cycle? Nope. So they don't do any of the things that would make something alive. And because they don't reproduce the way traditional living things reproduce, it's also very difficult to categorize them because we categorize stuff based on their level of relatedness. And because virus particles don't have relatedness to anything because they don't reproduce in a traditional way, we don't have any way to put categories on these things. So we talked about the two-part scientific names that we use for living things, the genus and the species. We don't have those for viruses. So there is a viral naming scheme, which kind of gets changed all the time. They change how they do the viral naming scheme. In general, it's shape-based. So we'll talk about these main ideas of viruses in a second. I'm going to jump around just a touch here. But they come in some general basic shapes. This isn't all of them, by the way. This is just a few of the shapes. But in general, we use this as part of the naming scheme for viruses. And I'm going to use COVID as an example of that, okay? So that's a coronavirus, and we'll talk about the shape associated with the coronavirus in a second. But it's named coronavirus. And then the second component of it is the iteration of coronavirus. So which one is it? There are a whole bunch of different coronaviruses, and this one just happens to be named after the year it was discovered. This is coronavirus 19. I didn't realize we were that close to the break. Okay, let's put this on pause, and I'll, right after the break, for you guys at home, we're gonna do a quick fire drill after our walk as well. So I'll see you guys at, normally it's 5 after, right? Is that right? So I'll see you guys at 11:15 then. All right, cool. See you in a bit. All right, guys, we're back. If you want to just say hello in the chat again, I got to take attendance for the second half. Did everybody make it back in? We didn't lose anyone? Okay. Cool. Thanks. Okay. Oscar, Dominic, Angela, Connor, Jane, Lauren, Sam, Elliot, Sarah, Lily, Sam, Clay, Sydney, Sharp, John. Okay. Okay. Okay, let's get back to it. So we were talking previously about the prokaryotes. So those are the single-celled organisms, really simple inside, two main types, bacteria and archaea. All right, so that's two of the kingdoms. Now we're going to take sort of, like I said, a side trip into the unclassifiable. We don't really sure where to put these guys. So, are they alive? No, not really. I guess if you're a virologist, you might say that they're alive. They kind of mimic the behavior of living things by hijacking living things, but they're not really alive by themselves. This is kind of some weird side topic. It's important to talk about it in taxonomy because we do have to do something with them. But so we were talking about how we name them then, because they don't have heredity like a normal reproducing organism, sexually or asexually. So we normally name them based on their shapes. Coronavirus, the current coronavirus, COVID-19, is named based on its shape. Corona, that's where the COVID part comes from, coronavirus. And 19 is just the year of discovery. So there are lots of other coronaviruses. You've probably had, I would say, several coronavirus infections over your lifetime. That usually just causes a cold. So you probably had lots of cold virus infections, I would assume. So, and a lot of them are less severe, and some COVID-19 infections are less severe. So it's not universally the same for everybody, but obviously this particular coronavirus strain can be significantly worse than a lot of other coronaviruses, but they all have the same shape, so it's defined by shape. So, and we'll get to the shapes in a sec. So they are non-living technically. And then they infect cells. So they get up inside cells. They cause illness. They don't all cause illness. So most of them do. But there are a number of viruses that that asymptomatically spread. You wouldn't know that you got them. They're probably moving in and out of organisms all the time and being communicated between organisms. But they're so small that if they are asymptomatically spreading between organisms, we don't really investigate that because we have no reason to investigate that. We have no reason to look for a particle if it doesn't do anything. So there's probably lots of virus particles that are kind of moving in and out of organisms that we just don't investigate because they don't do anything. So, or at least no visible damage. Because they operate by injecting their DNA into your DNA, often they take a piece of their DNA and actually plunk it into your DNA. They can potentially cause harmful mutations. You guys may have heard of, you may have got the shot for, it's supposed to prevent uterine cancer. What shot would that be for? Ladies, can you help me out on this? You may have gotten the shot when you were in elementary school. You get it for free? HPV, thank you, HPV vaccine. So you might have got the HPV vaccine, and now more and more they're giving it to males as well, because males obviously can infect females with HPV. So, but it doesn't cause uterine cancer for males. But the reason why it causes cancer is because of that change that it makes to the DNA. So it could potentially put a piece of DNA in the wrong spot and potentially cause something carcinogenic to occur. So viruses do have that ability. There's probably other virus infections that cause all kinds of stuff that we just don't understand. So this is a really like a burgeoning field in biology. We only recently discovered, like 30 years ago, DNA and how DNA worked. So like a lot of this stuff is not well understood. There's a basic format. So you got to have some genetic material in your virus. It's DNA or RNA. Did you guys talk about RNA last year in grade 10? A little bit maybe, about how it's like used to build proteins, maybe. We're going to talk about the beginning of the next unit anyway in genetics, but both of those are information containing molecules. So DNA would be the one that you have. Some viruses have RNA, and they use that RNA to build a piece of DNA and insert it into the organism's DNA that they're infecting. So both of them work the same way, but you got to have information inside your virus. So one of those has got to be in there, DNA or RNA. And then it's got a little capsule around it called a capsid. And their main mode of operation here is they inject themselves into a host cell and then they reproduce. So they get the cell to build copies of it. That's the basics. Viruses infect all living things. So not only animals, but plants, protists, fungus. There's viruses that infect fungus, and there's viruses that infect bacteria and archaea as well. So ones that infect bacteria are called phages. There's a lot of cool stuff you can do with phages. For example, you could potentially create a virus, genetically engineer a virus to infect a bacterial illness. There's a lot of current research in that area because some antibiotics are becoming ineffective. So another option potentially used to treat bacterial infections is to create a virus to kill your bacteria for you. It's kind of a cool idea. And so there's a fairly large area of research working on bacteriophages. So you'd intentionally infect somebody with a virus that would kill their bacterial infection. It's kind of a cool idea. There's two main life cycles for a virus that we're going to talk about, the lytic and the lysogenic cycle. We'll go into those in a second. And then there are lots of viral illnesses in humans. One really important thing to note about those is that we don't have any antibiotics to treat viral infections. So viral infections, in terms of how to treat them, is symptom treatment. How do you treat like a COVID infection? You treat their symptoms. So you give people things for a cough. You give people things to help open their airway. But in terms of actually treating the virus itself, antiviral medications are not great. They don't work super well. They can inhibit viral reproduction a little bit, but it's nothing like an antibiotic. So viral treatments are fairly weak. So we don't really have great treatments for viral infections, and that includes all of these. Although I must say, in recent years, especially for things like HIV, treating AIDS, there have been huge advances in reducing the number of HIV particles in people. So that's not, I guess, across the board true about viral treatments, but in general, we don't have great treatments. And certainly, antibiotics do not work. So an antibiotic will do absolutely nothing for a viral infection. If you have a cold, it's usually viral, and you take an antibiotic, it is completely ineffective. It will do absolutely nothing to your viral infection. So even though people ask for them. The one thing we do have, the big thing that we do have, is the potential for vaccination. And we're going to talk about how vaccinations work towards the end of this note. And I know everybody's hoping for COVID vaccine. That's there's certainly a possibility of that happening. So that's always like the silver bullet that people always look for viral infection is a potential vaccine. They typically work extremely well once you can get them to work. Okay, so viruses, non-living particle. They're surrounded by a capsid. We'll look at the shapes of those capsids in a second. There's no cytoplasm. There's no goo inside there. They're too small for that. They're about 0.1 micrometers in diameter. So let me show you a size scale comparison here so you can see how big that is compared to a typical cell. It's extremely, extremely small compared to a typical cell. I used to have a picture of this in the note. I don't know why I took it out. Here we go. That's not a great one. Show me a better one. That's pretty good. Oh, but it doesn't show red blood cell. I want a comparison to human cells. Give it all to me. Here's a good one. Perfect. Okay, so red blood cell. Did you guys get a chance to look at these in grade 10 under the microscope? Okay, so when you look at a red blood cell under the microscope on high power, okay, that's 400 times magnification for these, it looks like a speck. That's it, okay? They're really small. They are the smallest cells in your body. They're teeny tiny little cells. They have to be tiny because you have to squeeze them through your capillaries, through your circulatory system, so they can't be very big. So your red blood cells are real small. Next to this red blood cell that's in the corner there, which looks giant on this diagram, you can see an E. coli bacterium, okay? So an E. coli bacterium is about 1/10 the size of that tiny speck under the microscope, all right? So they are really small compared to human cells. Remember that the red blood cell is the smallest one. So they're really small. And then compared to an E. coli, if you start looking at virus particles, you can see that they are 1/100th or sometimes 1/1000th the size of a bacterium, okay? 0.1 micrometers. That is a teeny, 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 tiny little thing. All right? Way, way beyond the resolving capacity of our microscopes. Okay, you usually can't see viruses under a light microscope. You need an electron microscope to see viruses. They're very small. So you can see, I mean, basically all it is like a tiny little capsule with some DNA inside it, and that's it. Okay, there's no machinery in there. There's no cytoplasm. Let's see. Oh, I was hoping there would be a coronavirus on here, but there isn't. They're also very small. So they'd be about the same size as this rhinovirus here, about 30 nanometers. Okay, teeny, teeny, tiny. And because they are that, they are literally a piece of DNA in an envelope, they don't do anything by themselves. So that's important to note. Nothing happens on their own. They're just floating around. They have no metabolism of any kind. They can't process energy. They can't make waste. And they got a little piece of DNA or RNA in there. However, if they get into a living cell, and how they get into a living cell is specific to the virus type. So, for example, viruses could be specific to maple trees, or specific to tobacco plant, or specific to pigs, or whatever. Once they do get inside that particular host, they can potentially take control of the cell. So it's like a floating set of instructions, and the instructions are how to make a copy of it. That's all. Okay, because they're non-living, we already mentioned this, very difficult to categorize and place into phylogeny, because we can't really follow their historical genetics. So all we have is the current iterations of these things. They're specific to host. I mentioned this already. So they usually have a single host. However, some of them are multi-host. So a great example of a multi-host virus is rabies. Rabies can infect every mammal, certainly. I think rabies is limited to mammals. I have to double check this. But that's a lot of species. There's a lot of species of mammals. Usually viruses are not that flexible that they can just infect all different kinds of things. I'm retreading ground a touch here, but they're not alive. They don't do any of the life cycle stuff. The video that you're going to watch after this talks a little bit about the emergence of new viruses. Usually when we talk about a virus being new, we just mean new to humans. So it's possible that the virus was floating around in a population of bats, or floating around in a population of sea otters, or I don't know, something. And then there was a incident where it crossed the species boundary. But the very first viruses probably originated as parasitic cells. That's a pretty good guess. There are a number of types of bacteria that are parasitic, and some of them can operate even if there's cell membrane ruptures. And a lot of the precursors of a virus are sort of evident there. So there's a good chance that that's where they've originated. But to be perfectly honest, we don't have any evidence for any of this stuff because, again, there's no fossil record for viruses. So we don't really know. It's a bunch of guesswork. guys a second to write this stuff down. So, as previously mentioned, viruses cause all different kinds of illnesses. Some of them are very mild, at least for humans. Some of them are very severe. Some of them your body can completely eliminate, like a lot of adenoviruses, things that cause colds. So you can completely eliminate those viruses from your system. And some of them leave their DNA in your cells and then go on vacation. Those ones are a little bit more problematic, like the chicken pox. So if you've ever had chicken pox, you didn't get rid of it. You still have it in some of your spinal nerve roots. So some of that DNA from the chickenpox virus, varicella zoster virus, is still chilling back there. It's not active, so it's not reproducing virus particles, but it is hanging out in your DNA. And sometime later in your life, it's possible that that virus reproduction can occur again. So if you have a strong immune system, oftentimes your immune system will immediately recognize it if it starts to reproduce again and remove it, in which case you won't get chickenpox again. That's why you usually only get it once. However, if there's a period in your life later, like let's say you're in your 60s or 70s and your immune system isn't as strong, or there's just another part in your life where you have a weakened immune system for some reason, you can experience a second outbreak of varicella zoster, of chickenpox, which you usually get as shingles. You usually don't get full chickenpox again, you usually just get shingles. But again, it's still painful. So, but you don't ever get rid of it. The viral DNA is in there somewhere. Same thing is true for herpes virus, simplex 1, simplex 2. Once you get it, it stays in a facial nerve root pretty much forever. So you can get reemergences of the virus particles. Usually the more times you experience it, the stronger your immune response becomes, and you don't get them quite as often or quite as badly. Sometimes people only get 1 or 2 outbreaks, and then their immune system kind of gets on top of it, and you don't get them anymore. But some people experience them like for the rest of their life, and some people, they get more serious over the course of their life depending on your immune response. So those viral infections you can never get rid of. So there's all different kinds. They also in nature have an important ecological role. Oftentimes they act as population controls in nature. So when populations get very large, like you have too many ants in a rainforest or something like that, often a viral infection will cause a reduction in population. It prevents them from taking over. The higher your population density as a species, the more likely that you're going to get communication of virus particles between the individuals in your species. And that's true for humans too, by the way. It's one of the reasons why our current situation is so much different than a viral outbreak 100 years ago or 200 years ago. People move a lot more. People are in more dense populations on Earth. And so we have more virus communication between members of the population. We have to wear masks and things like that to try and limit the communication of those virus particles. So I'm going to get you guys to do a little extra research on that. Not coronavirus specifically, but on pandemics and epidemics. So there's a little bit of extra terminology I want you to investigate here. So you hear these words all the time: epidemic, pandemic, and then the last one that I want you to look up here is endemic. So what do those mean with regards to a viral infection? When does a viral infection become an epidemic? When is it a pandemic? And when is it endemic? These things are all related to each other. And then, sort of as an aside here, what is the status of the coronavirus? Is it an epidemic? Is it a pandemic? Is it endemic? I'll get you to come back to that in a minute, okay? So here's some examples of some viroid particles. This is the one that you see on the front of textbooks all the time because it looks cool. Look at it, it's like a spaceship. It lands on things, it's got these cool legs. And then it injects its DNA. It actually kind of like sits down on top of the cell membrane and actually pushes that little particle of viral DNA into the host cell. Cool. This is a bacteriophage though. So that shape of virus particle only infects bacteria. So you're never going to experience an infection from one of those. It doesn't work with animal cell membranes. So you can't get a piece of DNA across with one of these, which is one of the reasons why there's no overlap. Like things are very species specific. So that thing's not going to give you a viral illness. However, if we found a bacteriophage that could kill chlamydia, and you got chlamydia, we could potentially give you some of this. We could put it in your blood, or maybe find a way for you to consume it orally, although that would be harder to get broken down in your stomach acid, but probably be an injection. And then these could potentially destroy the bacteria in your blood. So it's kind of a cool idea. Most viruses that infect humans are coated in what's called an envelope. Okay, so that the envelope is like this part on the outside. And it often has proteins on it. So when those virus particles come in contact with your cell membrane, those particles, those proteins on the surface, are usually the same proteins that are already on the surface of your cells, of human cells. So when that touches the cell membrane of your cells, the cell says, oh, this is just a little piece of my cell membrane. And it'll just pull it into its own cell membrane. So as it pulls it into its own cell membrane, it pushes this particle inside the cell. That's what it does. It basically absorbs this particle when the particle hits it because it thinks this is supposed to be part of its cell membrane. It's very specific to these proteins that are sitting on the surface of the virus particle. And that is why you get something that's very specific to humans. It'll only have particles on the outside that human cells will recognize as their own. There is some overlap. So some species have a lot of similar proteins on their surface to humans. Pigs are a great example. Pigs have many membrane surface proteins that are similar to human membrane surface proteins. So there's quite a few viral illnesses that are communicated between pigs and humans. What's the one that people always talk about? Swine flu. Swine flu. So humans can get swine flu. Obviously pigs can get it. But there are similar membrane proteins on both of those virus particles, and so they can actually transfer between humans and animals. But oftentimes they're very specific to species. You also notice there is like a little sugar capsule or something, and then it's surrounding some viral DNA in the center. That's this little doodle in the middle, okay? DNA or RNA. In the case of HIV, HIV is called a retrovirus because it takes its RNA and it uses your body's cells to build its own DNA to match the RNA and then insert it into its genome. So the same thing with herpes simplex virus, chickenpox, same thing. Those are retroviruses. So they inject their DNA, or their RNA is converted into DNA, and that DNA gets put into your genome. It's a permanent thing. So if your cell that has that viral DNA in it makes a copy of itself, just like cells normally do, they do mitosis, it's actually going to make a copy of the virus's DNA as well every time it makes a copy. Because it just thinks it's part of its own DNA. And then it gets passed on and on and on. Here's an example of tobacco mosaic virus. This has a capsid on the outside that is specific to tobacco cells. So tobacco cells recognize that shape and say, oh yeah, that's just part of my cell membrane. And then it brings it into a tobacco cell. So it's specific to tobacco. This is an adenovirus, mostly affects humans. This is a coronavirus. There are several of them. Oh, by the way, the reason it's called a coronavirus is because it has this crown, this corona, of proteins that go all the way around the virus capsid. That's the corona in the coronavirus. It's like a halo that goes around it. under the microscope. Pretty simple little guy though. That's all it is. Just a little piece of RNA in the middle. So as I mentioned here, when the virus comes in contact with the cell membrane, basically the cell decides, oh, that's supposed to be in here. It'll bind to some receptors that are on the surface of the cell, and the cell will either open up the virus particle and allow the RNA to come in, or it will take it in as part of a vesicle like this. It'll vesicle it, bring it inside. And that's how the virus DNA gets distributed inside the cell. I think this just says exactly what I just said here. Yeah, they all have RNA or DNA with an envelope. The host wraps its own membrane around it. And the membrane is used as a disguise. That's the other thing. When these particles are floating around in the body, one of the reasons why the immune system doesn't immediately recognize it is because the proteins on the outside are proteins that look just like the proteins on your own cells. So when your immune cells see them, they just say, oh yeah, that's just whatever, this particle that's supposed to be in here, no big deal. Unless your immune system already knows that that particle isn't supposed to be there. That's what a vaccine does. We'll talk about that in a second. Those shapes that are on the outside of the virus, these guys right here, I'll go back down in 1 second. These are called antigens. An antigen is just a shape. Bacteria have antigens on the outside too, by the way. That's how your immune system recognizes them. So it's not just viruses that have antigens. Oh. I like that. All right, let's talk about vaccines. The magic bullet. The thing that we've all been waiting for. All right, let's imagine we've got a virus particle. It looks like this, okay? So there's your virus particle. The virus particle has got a whole bunch of shapes on the outside, okay? Those are antigens. When it first begins to start an infection in your body, okay, and the antigens, and it's replicating, and then your body's immune system figures out something is wrong. Something is causing cells to lyse. Something is causing new chemicals to appear in the blood that don't belong there. What happens is that your white blood cells start searching for an antigen. What are the shapes around here that don't belong? Okay? So your white blood cells will go around and say, okay, this is the wrong shape. this shape doesn't belong here. You've amped up the amount of white blood cells, and now they're searching all over. I mean, they do that anyway. They're crawling all around the inside of you right now looking for wrong shapes. But you amp up the number of white cells that you have when stuff starts to go wrong inside, okay? It activates an immune response when cells start popping all over the place. And now your white blood cells are looking everywhere for shapes that don't belong. When they find a shape that doesn't belong, like this orange antigen right here, the white blood cell will start to build flags to go on that shape. It has a sticky end. It has a little sticky end over here. Looks like a Y. And the Y, the sticky end, only sticks to that shape. It's built specifically only to adhere to that specific shape. Okay? And then it builds a lot of those flags. Another type of white cell called a T cell starts building a whole bunch of flags and releasing them out in the blood. And those flags go around. I like to think of them as bad guy flags. And then they stick on to those antigens of the virus particles that are floating around. Sometimes that's enough. So if it's a bacteria, sometimes if you stick a bunch of flags to their antigens, the bacteria can't reproduce inside of the cell, and that's enough. It inactivates the virus or the bacteria just from sticking a bunch of stuff to it. Not always. The other thing the flag does is it tells other white blood cells, phagocytes, to eat that. It's like, hey, this is a bad guy, eat this. So the white cells will then cruise around and look for things with the flags on them and eat them. That's how a typical immune response works in your body. And the real delay is how long it takes before you start making flags. So it's a long process. Your body's got to figure out, okay, what's the wrong shapes? Then it's got to say, okay, let's make a flag. This is how you build the flag. Now we got to build the flags. We're talking about days, okay? So it takes 3-5 days for your body to start making the flags, get millions of flags, billions of flags, and now it's at a level where it starts being functional and actually inactivating bacteria or viruses. It's getting things to eat those virus particles or getting things to eat those bacteria. These flags, by the way, can also be put outside of your cells that have a virus inside. Also very useful. So you can stick it on a cell and say, okay, there's some virus in this cell. I'm going to put a flag on it. And then your white cells will actually eat your own cells that have virus inside to prevent them from filling up with virus and exploding and reproducing the virus. Okay, great system. But the main problem here is how long it takes you to build flags. A while. Like I said, there's a big delay. And by the time you get that whole system going and start building flags, you're sick. You've got the full illness, whatever this happens to be. So how a vaccine works is instead of giving you the virus, which would be, obviously you don't want to get ill, what it does is it gives you just these. Hey, let's just give someone a bunch of these. When those show up in your blood, your white blood cells say, oh, that's a weird shape, that doesn't belong here. And it starts building the flags. So you get the ability to start building the flags, even though you don't have the viral infection or bacterial infection. That's the key. So when this thing shows up, you already can build the flags. It's a very short turnaround time. Your white blood cells will recognize it, and then immediately you already are like, okay, release the flags, we have flags for this, release the flags. It's pretty instantaneous. It may be like a 12-hour turnaround or something, but you start really releasing those flags very quickly. So those flags are called antibodies. You may have heard of this before. Your body's antibody system builds bad guy flags. Question?
Do the flags have like an expiry date? Can it last for a while?
They stay in your blood for a little while, but the key is that the cells that know how to build the flags, you keep some of those. So those T cells that know how to build those specific flags hang out in your bone marrow. And so if you do need them again in the future, you can start reproducing those cells, and then they will produce a whole bunch more flags. And it's a very quick turnaround time as long as you have them in your bone marrow. Depending on the type of flag, sometimes you keep that immunity for a really long time. 7 years, 10 years, 20 years. Sometimes the immunity is much more short-lived though. So depending on the type of flag, Sometimes you will only keep your immunity for a year or 2 years. So that one, like for example, you guys have had tetanus shot. So tetanus shot tells you which flags to build for tetanus, but it only lasts about 7 years. And after about 7 years' time, those T cells kind of start to die off in your bone marrow, and so you need an update to your vaccine. You get another vaccine, it does the whole process again, and then you get a strengthened immune response. So it's not perfect. The other problem with bacteria, by the way, this works great. So usually when we can figure out how to create a good bacterial vaccine, they tend to work really well. Bacteria mutate, so they change the shapes on their outsides fairly quickly. But a lot of bacteria have parts on the shapes, antigens, that are always the same, no matter which iteration of that bacteria you get, even though it's evolved and changed and stuff, it still has similar shapes. And so if you have a good vaccine that really captures those consistent shapes, usually you can use it for a long time. So we have a lot of vaccines that are very good. Chickenpox vaccine, oh no, that's viral, what's a good vaccine that we have for a bacterial infection? I'm gonna think about this one for a second. I'll come back to that. I gotta think about that. But anyway, the problem with viral vaccines is that viruses mutate very, very quickly. So the shapes that they have, the antigens on the outside, are often very different from one generation to the next of a virus, which is why for lots of viruses we don't have vaccines. It's very hard to figure out which parts of the virus are always the same between every iteration. And so you talk about how many COVID vaccines are in development. 200? There's a lot of different people working on different vaccines. When we talk about there being different vaccines, primarily what we're talking about is that they're using different shapes. They've chosen a different selection of shapes that they're hoping these are the shapes that are always going to be the same on the coronavirus, and hopefully those will be the ones that'll initiate an immune response and potentially protect you from coronavirus. But like I said, a lot of viruses mutate very quickly. The flu virus is a great example of a very fast mutating virus. So the flu vaccine is usually good for 1, maybe 2 years. But because the flu virus is constantly mutating, you really do need to get a new vaccine every year, because the vaccine every year is for different shapes for the flu virus that year. That's what this year's shapes look like. And next year's shapes might be a little different, and you need a different vaccine. There's a very strong possibility that that is how the coronavirus vaccine will work, if we do figure out how to get one to work. There's no guarantees on that, by the way. But if we do figure out that a way to make a vaccine work, there is a strong possibility that you have to get one every year because it may mutate fairly quickly. So some things to consider. Anyway, that's the basics, okay, for vaccines. So they are either a mixture of a weakened form of the virus, or they are straight up just parts of the virus. They don't usually give you the virus. In the olden days, when vaccines were first invented, like 200-300 years ago, they are that old, which is crazy, they used to just give you the illness, but usually a weakened form of the illness. So for example, to get the smallpox vaccine 200 years ago, they gave you a different viral illness that was very similar to smallpox, but a lot less deadly than smallpox, called cowpox. So they just gave you a different illness, which is kind of weird. You still get sick, but only like a little sick, instead of smallpox, which is often fatal. Smallpox is terrible. So that's one way of doing things. Not a great way. Now, we actually have bacteria build the shapes for us in the vaccines, which is crazy. So we reverse engineer it. We figure out the DNA of the virus. We put some of that DNA, not the whole thing, but parts of it into bacteria. And we actually have the bacteria manufacture the vaccine for us. Cool. Let's use these microbes against each other. That's the new way. It works extremely well. You also, by the way, when you make a vaccine, don't want to make one that triggers too strong of an immune response from people and kills them. So that's also problematic. You don't want people to be strongly allergic to your vaccine that you're using either and potentially kill people. So that's one of the reasons why you got to test these things. You got to test them for severe allergies. You got to test them to make sure that they cause an immune response. You got to test them to make sure that the immune response actually prevents the infection from the virus, which is why large clinical trials are important before you just say, oh, I got a vaccine for this, and you just start injecting it into people. Which is why I'm very, very dubious about claims that the Russians have a functional vaccine. I think it's highly unlikely that it's proven safe at this point. They're pretty much just guessing. Not a good idea in my opinion, but I suppose if you're in Russia, you have to decide whether you want to take that risk or not. Wouldn't be my first choice. Whereas here we have a lengthy process. Thousands of people get the vaccine. They check for efficacy and they check for allergic reactions. I'd feel a lot more comfortable if there was a vaccine approved here.
How would you know the long-term effects it has? Like, you're never really gonna know that until.
Difficult to say. Difficult to say. So we usually base it on previous vaccine. We do have long-term health information for a lot of vaccines. So if the vaccine works on the immune system in a similar way, we make some assumptions about long-term health effects. But you're right, there's no perfect answer for that one. We don't have long-term data usually, and certainly not in a situation like this where we don't have a long time to produce a vaccine.
And isn't it like pretty unlikely we're gonna get a vaccine based on like the fact that not many people have antibodies after like 3 months?
Is it what, sorry?
I heard somewhere that it probably wasn't likely we're gonna get a vaccine because even after 3 months, like a fraction of people who actually have COVID even have antibodies to it.
Difficult to say. There have been some cases of reinfection. Now, it really depends on how you initiate the immune response. There's different ways to initiate it. And sometimes getting the infection itself is not as effective at protecting you from a second instance of the infection as a vaccine can potentially, depending on how the immune system is activated. But is it possible that none of these vaccines will be effective because of that reason? Yes, it is. Yeah, absolutely. So we are, operating on a hope model. There's definitely no guarantees when it comes to vaccines. Oh, go ahead, Mohammed. What's up? Oh, that's an interesting question. So Mohammed just asked why some people have an association between vaccinations and autism, which is a really interesting question. So there was some research done 15 years ago that showed that there was potentially some connection between autism and vaccines, but subsequent to that, we found out that scientist faked a lot of data that was in that study. And so even though that study has been retracted and that there is no further evidence to show any connection, even though there have been many studies looking at that connection since then, that idea still persists. So you can't take the idea back from the public once you put that out there, which is unfortunate. That person caused a lot of damage and is, I don't think, employed anywhere anymore. Yes?
Are there any vaccinations that you only have to get once and then never again?
Great question.
Because I know for MMR, you get it when you're really little and then you get it again when you're 4 or 5. But
Yes.
you don't ever get it again.
I don't know if you ever need an update for those. Measles, mumps, and rubella. A lot of those illnesses are not as serious when you're older. That is a great question. If you ever need an update to MMR, can I investigate that? I'll get back to you on that one. I'm trying to think if there's one where you get a vaccine and you never have to get an update for it. That might be an example. Let me figure that out. I don't know the answer to that question. That's an interesting question. I'm going to get you guys to do a little bit of background information into our immunization schedule. So you guys have probably had vaccines. They are required for you to attend school. So you've probably had a few. I'd like you to look into what are the vaccines that people have to get. You might not even know the ones that you've received. It's really cool that a lot of vaccines exist now that didn't exist when I was a kid. I got chicken pox and it really sucked. And maybe when I'm older, I'll get shingles. But you don't ever have to get chicken pox or shingles if you don't want to. You can get the chicken pox vaccine and it actually works pretty well. I think it has like a 75 or 80% efficacy rate. Hooray! So you don't have to get chicken pox. So I just want you to do a quick little investigation. This is going to be in a moment here. What can you actually get a vaccine for, and what do you get vaccines for? You might be surprised to see what we can actually get vaccinated against. It's a lot. We vaccinate, well, not here in North America, but in Europe, they vaccinate chickens for salmonella. So you can't get salmonella poisoning from eggs rather, in Europe because they vaccinate their chickens for salmonella. The reason that we don't do it here is because it's expensive. That's not honestly, I think that's the actual reason. We don't do it here, and because of our supply chain for eggs here, we refrigerate our eggs. But in a lot of European supply chains, they don't refrigerate eggs. So they don't wash and refrigerate eggs in Europe. They just use the eggs that come out of the chickens. They actually last quite a long time just on a countertop. Not quite as long as they do in the fridge, but it's quite long. Long, probably for most people. But in order to do that, you do have to make sure that they don't have salmonella. So you can vaccinate chickens. Yes?
Wait, so does that mean that if you put your eggs in the fridge, it'll get rid of the salmonella?
It doesn't get rid of it. It prevents the growth of salmonella by keeping them in the fridge. Yeah. We wash our eggs here. So eggs have like a natural like waxy protective coating on the outside, which allows them to keep really long in room temperature. But when you wash the salmonella off the outside, it actually removes the wax coating too. So you have to put them in the fridge or they go bad really quickly. So here we have to put our eggs in the fridge. Anyway, you can skip those questions at the very bottom. I put a line through that. Don't worry about that stuff. Yeah, you don't really need to know about mumps, but I would like you to know what vaccines are available. So I think I'm going to give you some time to do this right now. There was another question I asked you to look at too, which was up here. Oh yeah, the difference between an epidemic, a pandemic, and when something becomes endemic. What does that mean? It's usually applied to viral illnesses. And then I'm going to bring you guys back at, why don't I give you 15 minutes, okay? I'll see you at 12:20 for those of you guys that are at home. Okay. Okay. Sorry, I realize not everybody's done all that stuff yet. So I gotta leave something for you to do during Learning Block 3 anyway. So just a quick reminder that for each of these sections, oh, is this gonna crash again? Bleh. For each of these sections, there is like a couple little supplementary videos to watch along with them too. They're really quite good. This one on where a new virus comes from is really cool. So it talks about like a bunch of pretty good educated guesses about where the coronavirus probably came from, and in general, how some past outbreaks have been communicated from other species to humans, which is interesting. And then there's a great one for the protists coming up too that you guys should definitely watch. So okay, so let's talk about protists. So protists are the weird kingdom. Like by definition, they're the weird one, because if you look at like the phylogeny of all of the modern kingdoms, okay, so like you like follow up the little, the little phylogeny tree, right? And you'll get ones like, okay, animals fit here. That's clear. And then we'd be like, okay, well, plants fit here. And like all of the prokaryotes fit here. So there's two kingdoms in that one. And then it's like, okay, fungus fits here. And then there's like a whole bunch of other ones. There's like this and like two that fit over here. If you actually like look at the phylogeny, there's like a whole bunch of other stuff. Most of these are single-celled. They have names. I don't know what all of them are off the top of my head, but there's like a bunch of other groups. And it's like, okay, well, these don't fit. They're not plants. They're not animals. They're not fungus. let's just put them in another one. Just kind of squeezed them all together into another kingdom. So we took all these things that are phylogenetically not really related to each other. They're like kind of quite different from each other. And we just squeezed them all into a separate kingdom. We took all those branches and we just said, all right, let's just put them all over here and we'll call that Protists. So that's what we did. Okay, so this is the real weird one. When we talk about this group and we talk about like what makes something a protist, like what does a protist look like and stuff, it's like, that's hard to say because they are quite different. Most of them are unicellular. It's definitely the most diverse of the eukaryotes. They're all eukaryotic, by the way. So they all have a nucleus and Golgis and like all the things that make you a eukaryote. Almost all of them are aquatic. So I think that's part of the reason why historically we've categorized them all together. They mostly live in water or in very wet environments. So these are like water organisms. They are often producers, so a lot of them are producers, but then many of them are also consumers. Some of them are both. So some of them do photosynthesis and they eat things, which is weird. So they're like hybrids of both of those things. Some of them are responsible for diseases, so some of them are pathogens. Very few are pathogenic, but some of them are. For example, the protist that causes, there's a protist that causes malaria. That's actually a protist that can live inside of cells, so it gets inside of your cells. It's kind of cool. As I mentioned, they're all eukaryotes, so they have nuclei. We're going to talk about endosymbiosis in this one? That might be towards the end. I'll come back to that point. Right. So it is a grouping that is just all the other ones. What doesn't fit into the typical kingdoms? Protists. Here they go over on this side. So difficult to define them. They get around in various ways, although not all of them do. So some of them are stuck in one place. A great example of that would be seaweed. So seaweed is actually a protist. It's not a plant. And it doesn't go anywhere. It acts a lot like a plant, so it doesn't move around at all. But a lot of the other ones can move. So they have locomotion. They have a whole bunch of different ways to get energy. Like I said, some of them do photosynthesis, some of them eat stuff. They have lots of different methods of reproduction. Some of them do straight up mitosis. So they will just grow, split, grow, split. Some of them will bud, which means that they build a copy of themselves beside themselves, attached to themselves, and then just push it off, and then it becomes another thing. And I'm talking about multicellular organisms. So it'll build a complete multicellular copy of itself that's stuck to it, and then it'll just bust it off, and then it'll become its own. So some of them reproduce like that. That's called budding. Some of them do sexual reproduction. So they sexually reproduce with each other. They have some kind of egg and sperm system. Some of them do a weird hybrid of sexual reproduction where two cells come together and they form something in the middle of the two of them where they exchange genetic information. All kinds of stuff. Some of them are parasitic. Kelp was the example. That seaweed would be like a very big multicellular organism, and then many of them are single-celled. They're all relatively large compared to bacteria. So if you were to look under the microscope at these, even the really small protists, like the ones you'd find in swamp water or whatever, they're much bigger than a bacterial cell. So you can like see them swimming around. They're fairly large single-celled organisms. They're quite a bit bigger than your cells. So they're bigger than human body cells when you look at them under the microscope. I gotta see if we can find some way to do that. That'd be so cool. We normally do that. We normally look at them, but anyway, they zip around. Some of them are quite fast. We're gonna look at three in particular. These are the ones that we normally would look at in the lab. We might do just a little bit of research on these instead. But Paramecium, Euglena, and Amoeba are the three that we're gonna look at in more detail. In the past, I've had students make biological drawings. I think we're going to skip that. You guys aren't even going to look at them under the microscopes. That seems totally a waste of your time. So maybe we'll find something else to do for that instead. But I am going to get you to compare and contrast those three. Okay, so that's part of what we're going to do in this last little bit here. compare their shapes, compare their size, compare their abundance and movement and stuff like that. Not necessarily in a slide because we're not going to look at swamp water, but you'll do a little bit of research. Okay, you can skip this part. Review microscope skills. I mean, you're not going to use a microscope, so I'm not going to make you waste your time. That is usually part of this course. We use the microscope a fair bit, but we will have to figure out some kind of alternative to that. What we typically do is, because some of them zip around so much, you got to put some slow juice on them. So the slow juice is called methyl cellulose, and it slows their nervous system is what it does. It slows down their ability to metabolize and move their body parts around so that they zip around slower on the slide and you can see them. Because some of them are quite fast. If you're looking at them on the slide, they'll just go whoop across your field of view and you can't really see them. So you slow them down. These are the major groups, okay? Of protists. Does it say that at the top? I'm just going to write it again. Characters, these are the major groups. Oops. Otherwise known as phylums. Right below kingdom, phylum. It's the grouping right below in the taxonomic structure. So there's euglenoids, ciliates, apocomplexa, diatoms, amoebas, slime molds, and red algae. You don't need to know these, okay? So I'm not going to ask you on a quiz like what are the phylums of protists. There's so much diversity here that I don't think it's really fair to ask you to know all of this. So, but you should know that there are several different groups and they are very different from one another in terms of what they look like, how they do their metabolism, et cetera, okay? The three things that I am going to get you to know about is just, I would like you to know a little bit about each of these that I'm going to get you to find more detail on, okay? So the three species that you're going to find out more about are Euglena. What were the other two here? Euglena. What was the other one? Amoeba and Paramecium, right. There's a Paramecium right there, and there's an amoeba, because they are quite different. What I do want you to know about protists is the three basic types of protist. So of the protists, you can usually separate them into three basic classes of protists, okay? The first one, I'm just making a little table here. I don't know if you guys want to do this on like a Google Doc or something like that. This doesn't really fit into the notes package very well, but a Google Doc would be a good place to do this. So, like I said, in general, there are three main kinds of protists. The first one are the plant-like protists. And they're characterized as being like plants. That's why they're called plant-like protists. So these are pretty easy to identify because if it looks like a plant and it's a protist, it's a plant-like protist. If you keep fish at home and you've ever seen algae growing on the inside of your tank, you might have thought, oh, that looks just like a plant growing in there. But it's not. It's a protist. It's a plant-like protist. Or if you've ever been scuba diving and you've gotten like caught in the seaweed or like seen some seaweed waving underwater, you might be like, oh, that looks like a plant. But it's not. It's a plant-like protist. Because plants don't live underwater. So there's a dead giveaway for you. Some plants have components that live underwater, like a root system or something, but plants don't live entirely underwater. If it's totally living underwater, it's a protist. So plant-like protists don't usually move. That is not 100% true, but it is generally true. This is a tough one because there's a lot of exceptions to these because protists are so diverse. But in general, plant-like ones don't move. When we say motile, it means don't move to a new place. They may move, but they don't go anywhere. So that's motile. They're not motile. They're all autotrophs. So that's the one key thing that makes you a plant-like protist is, do you do photosynthesis? If you do it, you're a plant-like protist. That also means that they're usually green because they use chlorophyll to do photosynthesis. So they are usually green. With some exceptions. Red algae is also photosynthetic and a plant-like protist, but it's red. It has a different chemical that it uses to do photosynthesis, but still, it's still plant-like protist. So cyanobacteria are bacteria that do photosynthesis or something similar to photosynthesis. And along with cyanobacteria, the plant-like protists form the bottom of every single aquatic ecosystem. So they're absolutely pivotal to the survival of stuff in the water. If you've ever been like scuba diving in a reef or something, or even just swimming off the coast, I mean, you probably noticed that there's like a lot of plant-like things growing down there that are feeding everything in the water. So these are almost all protists. So the main groupings of these are green algae, brown or red algae, and euglena. Euglena are funny in that they can move around. So they do photosynthesis, but they actually just swim around. They're swimmy plants. Not a lot of swimmy plants out there. We good? Can I keep going? Yeah. Not sure the timing on this. Okay, so you got your plant-like protists, which are the most common. In the middle here, I've got the animal-like protists. So if it's a protist and it looks like an animal, it's an animal-like protist. Another word for animal-like protist is protozoan. You'll often hear them called protozoans. In biology, a zoan just means animal-like. It has the word zoo in it. Same prefix as zoology. Yeah, I believe he's at home, but yeah, I think so. I think he's in part of 4B. No worries. Sure, no problem. I'll just email him. Sure, no problem. Okay, all right, no worries. Bye. So they're motile, they move. They're all heterotrophs, which means they eat stuff. It's a short list, guys. That's it. They move and they eat things. That makes you an animal-like protist. And some examples, you're going to do a little more research into the amoeba, but the stentor is one of them. Paramecium, stentors are pretty cool too. Oh, I wonder if I could find a cool video of a stentor. I think I've got a video of some of these battling each other. It's kind of cool. Under the microscope, they eat each other, they fight and stuff. It's cool. I'll see if I can find a cool video of that. There's a lot of weird people on YouTube. I'm just imagining like people at home just being like, oh, I'm gonna videotape some protists battling. But I guess I'm weird too because I enjoy it, so. All right, and then the last grouping here is fungi-like protists. So they're not a plant, they're not an animal. These ones are like a fungus. Most fungi-like protists are slime molds. I'll put up a video of what a slime mold looks like too. It's this really cool, like pulsating mold that it actually, you can like see it pulse. It's not an actual fungus, so it's see if I can describe what they're like. They're heterotrophic, so they eat stuff just like fungus eats stuff. They are individual cells, but at a certain point in their life cycle, they send out this chemical messenger that causes all of the singles to come together and form a loosely multicellular organism. They're still separate cells, but they work together in a big colony. And that may actually be the origin of the original multi-celled organism, because it's like the first in nature that we've seen of like single-celled organisms working as a superorganism. And oh, I gotta show you a video of it. Anyway, it pulses around, whoop, 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 and it like sends out little tendrils as it pulses. And as it finds new sources of food, it will actually push all of the individuals down that little tendril, and then it'll grow in that new location over the food, and then it'll push out more tendrils, find more food, and it kind of like moves around as this like pulsing blob. So anyway, I'll put a video up of it. It's really cool. They're heterotrophs. They eat stuff like fungus eats stuff. They have, I think, the coolest life cycle of any organism.
Please excuse this interruption. Just a reminder that students can't be dropped off at Central Market by their families. We need to have students dropped off away from the school, but not on the property of Central Market. Also, bus students and people walking home. Keep in mind that you must socially distance when inside and outside of the school. You need to wear masks outside on the school bus, and once you get off the school bus to enter the school. Nobody should be congregating together while waiting to get into the school. All students must maintain 6 feet distance among one another. Staff and admin will be monitoring this daily, and we must reinforce the health and safety rules for COVID-19. Safety is paramount for our schools. So near the buses, there shouldn't be groups of people congregating when going on or coming off the buses. You need your masks on, and you need to socially distance. Thank you.
I think the school board is getting nervous too because the numbers are starting to go back up again quite a bit. So
There's huge clumps outside of the school. Everyone like clumps together.
yeah, the school board's worried about that because it
I
does.
was not happy about it this morning.
It only takes like a few cases and then it really like it spreads very quickly. So anyway, we'll keep going here. Right, they have a really cool life cycle. Like I said, they start off as individuals that kind of get called together into this superorganism. I'll post a video. I have a video of dog vomit slime mold, which is a good example of a slime mold. And they are motile. They move around. So it's like a fungus, but like a fungus that can crawl around. And it's made, instead of all the cells being the same genetically, they're actually genetically different cells, but just a community that works together as a single organism. It's really cool. So those are the fungi-like protists. Most protists fit pretty clearly into one of these three, although there are a couple exceptions, as I mentioned. So what I'm gonna get you guys to do for this is have a look in more detail. Now, I mentioned biological drawing for each of these. Don't draw these, okay? That's something that we normally do with the microscopes and stuff. Don't bother drawing these. You don't have to draw these. Find a good picture, okay? And by picture, I mean like a microscope photo, an actual photo of the thing. Don't find like a diagram, okay? The diagram's not gonna give you the good stuff. The whole idea of this is you can see what they actually look like under the microscope, and there's a million images of them actually under the microscope. So find a good image. And then what I'd like you to find out here, just do a little bit of research, this would be easy to find, is some just, what are the key characteristics of that organism? How do you know you're looking at an amoeba? How does it move? What are the organelles that you can clearly see on the amoeba under the microscope? Okay, what are its key characteristics and what does it do? Does it eat things? Does it do photosynthesis? Like, what is its key behaviors? So it's the same thing for these three. Normally you would do this and you'd look at them under the microscope and draw them, but you kind of got to skip that part. So this is a lot easier. You're going to find a good microscope picture and then just find out the key characteristics of that organism. And then finish it off by answering these questions in the textbook. Any questions about what you've got to do? Not really, eh? All right. Let me throw up here the day 3 stuff again and just mention that there are these videos that go along with it as well that you should watch. This one talks about the groups of protists in a little bit more detail, not a ton. And it mentions here what you got to do at home. Learning block 3, as usual, I will be here the whole time in the virtual classroom. I've decided to turn my camera off because it's a little bit weird that I'm sitting here on camera the entire time and I can't tell when students come in. So people are just like, could be watching me, just like, that's weird, that's weird. So I just have my camera off. I am in it, okay? I have my speakers on. So when you show up, just say hi, say something, and then I'll turn my camera on, I'll jump in with you. But please say something. If you don't say anything, I won't know that you're there. There's no way for me to turn that on. No questions about anything? You guys are good, eh? All right, day 3 in the bag. Tomorrow, we're gonna finish this unit. So crazy. We'll talk about what the project that you're gonna do at the end is as well, to sort of summarize all of this stuff. And then we're gonna start talking about genetics. Crazy.